Proteins. All right, so proteins are important, really important. They are amino acid polymers which make us what we are, right? So the fact that it says amino acid polymers means that these are polymers, you know what that means, made out of amino acids, and they're important, okay? Proteins have incredible versatility of structure and function, right? So your muscles are made out of protein, your blood is made out of protein, your hair is made out of protein, and all of those things are very, very different from each other, right? Your muscles can get shorter and then get longer, and without them, you wouldn't be able to move, yeah? Pretty important. Actin and myosin are the two proteins in your muscles that grab onto each other and pull and so on. Raise your hand if you remember how actin and myosin work from physiology class. Great, yeah, so actin and myosin are two proteins. They can change shape and grab each other and let go of each other very rapidly. And from being able to grab each other, pull each other, and then let go of each other, and then grab and pull again, that makes your muscles shorter, and that is hugely important. And the function of actin and myosin in your muscles, can you close your eyes and imagine little pictures in your textbooks of what actin and myosin looked like, right? Then hopefully you realize that the shape of those molecules helps them do their job, making your muscles contract. Likewise, your red blood cells, what protein do they have inside them? Hemoglobin is the protein. Raise your hand if you remember hemoglobin. Hopefully you remember the vocabulary word at least. If you remember the shape of hemoglobin, you'll see hopefully that that actually is vital to how it works too. Lastly, your hair is made out of a protein called keratin. It's a very similar protein, or it is the same protein as in your fingernails and so on. All right, so very different jobs, very different things, and yet all made out of the same stuff, a bunch of amino acids. And so when we look at the level of the molecule, proteins are molecules just like carbohydrates are molecules, just like lipids are molecules, yeah? Protein molecules they're all macromolecules, gigantic molecules, polymers, and they're diverse and complex at the molecular level, right? So each of these proteins has a particular shape that helps it do its job, yeah? Anyone remember from physio fibrin in the bottom left-hand corner? Where do we find fibrin? Dig into your memory banks for that one. How many people remember fibrin from physio? 2 or 3. What is it, Kelsang? Did you guys hear that? Is that correct? Is she right? She's right, yeah. So, is that from my class? Of course it is. Of course it is. All right. So, how many people knew that and remember that? How many people didn't remember it but remember it now? Okay, good. So bringing that up isn't wasted, right? So fibrin, it makes your blood clot, right? So a blood clot probably takes a cajillion of these fibrin molecules. Imagine a cajillion of those molecules that look like that all released in the same place. What would they do? They might get tangled up into each other, yeah? So the shape of these guys as unwieldy, gangly things that could get stuck on each other might, as a group, make a really good net, don't you think? Does that make sense? You can imagine a fibrin net being made out of little pieces like this, yeah? So the function of fibrin to clot your blood depends, at least in part, on the shape of the protein itself, yeah? Protein shape matters, just as we learned that the shapes of other molecules matters. Yeah, Michael, you have a question? Yeah, that's the stuff I think that platelets activate, or it's inside platelets. You would think I would remember that kind of thing, and I don't. But yeah, it's if you're thinking platelets when you think fibrin, you're thinking the right thing. All right, ATP synthase. Can we infer what ATP synthase does? based on its name? Makes ATP, synthesizes ATP, hence the name, right? And does it look like anything? So hopefully when we talk about chapter 5, yeah, what does it look like to you? It looks like a medal. It does kind of look like a medal for valor or for meritorious service, doesn't it? It's actually, though, it's a three-dimensional thing. And it's actually this thing, this red part, spins around. And as it spins around, it makes ATP. Every time it spins, does a full 360 on its little spindle axis, 3 ATPs are made, right? So knowing that, maybe, hopefully, you might think it looks like a generator or a dynamo of some kind, and it actually works very much in the same way. And again, the shape of the molecule is really, really important to the function of the molecule, okay? Protein function depends on shape. But again, these shapes are a lot more different than the shapes of carbohydrates. We saw 3 or 4 or 5 major carbohydrates, right? We saw, what were they? Starch. What else? Glycogen. Cellulose. Those are the three fundamental ones, right? All of them look like ribbons, yeah? And the ribbons were straight or helical, but they're all sort of linear and not particularly unusual shapes, yeah? We also saw chitin. That was also a filament. We saw peptidoglycan. We only saw 2 units of that, but that's also filamentous. These things, can you see that they're still linear? Look at rubisco in the top right-hand corner. Can you see that it's ribbon-shaped also? You see the little ribbons of blue, the ribbons of turquoise, and the ribbons of sort of a brownish color here? Right? So each of these little globs is a ribbon also. So they're linear also, but the shape of these aggregate ribbons is like a little flower, isn't it? The shape of these aggregate ribbons is whatever shape that is. You can see the ribbons in insulin, yeah? You can see the ribbons here generating a very complex shape in nitrogenase, right? So let's look again. There are ways of looking at proteins. Here's the same protein shown a couple of different ways. We have a space-filling molecule, or model rather. So this is the same protein on the bottom as it is on the top, yeah? On the bottom, it looks like globs. And the glob is made out of little balls, yeah? What are the little balls? Those are atoms in the protein molecule, yeah? Individual atoms, yeah? Just like we saw the individual atoms in the octane, yeah? But this is a much, much bigger molecule than octane, yeah? Octane is a single small molecule. This is a polymer, yeah? A macromolecule, so it's a lot bigger, but you can see the individual atoms in it, yeah? What about on the top? Well, we can see that this globular thing on the bottom is made out of clumps of ribbons, yeah? You see the ribbons there? Okay, so again, proteins are linear like carbohydrates are linear. Let's look at that. So first of all, proteins are amino acid polymers. Let's first learn about amino acids. Amino acids are made out of functional groups, right? On the left, we have an amino group in blue. On the right, a carboxyl group in orange. And in the center, a central carbon with a hydrogen on one side and a side group with the letter R. That's our placeholder, yeah? So all amino acids are the same in some ways and different in others. And there are 20 different kinds of amino acids, okay? 20 different kinds of amino acids, each with different chemical properties based on the green side group, okay? So this is one particular side group on the right. This is tyrosine, and it has a little OH, right? That OH can do hydrogen bonding, so that makes this guy a little bit polar. Does that make sense? All right, but there's 20 different kinds. They're all the same insofar as they have an amino group and a carboxyl group with a central carbon. They're all different insofar as they have a different side group. Okay? Is that clear? Right? So the amino group and the carboxyl group, we call that the backbone. The R group, the side group, we call that the side group, or the side chain, sometimes I'll say. Yeah? Guys got it? All right. So let's look at the 20 different amino acids. Some are polar, some are nonpolar. Which one's polar and which one's nonpolar? The one on the left is polar or nonpolar? Leucine in the sky with diamonds. It's polar or nonpolar? Nonpolar, right? It's only carbon and hydrogen. Those are nonpolar. What about the one on the right? Hydrophilic, in other words, polar. Yeah? All right. So we can buy some as nutritional supplements, right? We have L-lysine. My dad sometimes buys this. And then here's the pirate amino acid. Sorry. Arginine. Sorry. All right. So that is a joke from my SF State biochemistry professor from over 10 years ago. So anyway, so here we go. Here's all 20 in a little chart, right? Some are non-polaire et hydrophobe. Yeah? Yeah? Some are acide. Some are basique. Yeah? Some are neutre. Right? Hopefully, even if you don't speak French, as I clearly speak fluently, that you can still suss out the meaning of this, right? Sometimes when you rip your stuff off the internet, you rip it from websites that are in French. And then you get to pretend you're French for a few seconds. Here's the one from your actual textbook, organized in the ways it sees fit. All right, so 20 different amino acids, yeah? So how do we, but a typical protein is made, I think the median number of amino acids of a typical protein is around 300 to 500. So hundreds of amino acids to make a particular protein, sometimes thousands. How do we get them to form together? Well, dehydration synthesis, just like lipids, just like carbohydrates, yeah? The OH group, that's a subset of our carboxyl group on the one side. The H is from the amino group, the amine group, on the other side of the other amino acid, right? So here really is our best comparison to the rail cars, yeah? We have on the left the amino group, that's like the pin, and we have on the right, that's like the ring, and every amino acid has a ring and a pin, or an amino and a carboxyl, just like every rail car has a ring and a pin. You understand? Guys get that? All right, they link together. Once that bond is formed, it's a bond between a carbon and nitrogen atom. It's called a peptide bond. It has its own name because it's important. And there's many kinds of peptide bonds, but they're all basically two amino acids stuck together. Okay? All right. So short peptide chains are also found linking peptidoglycan strands, right? So here are some amino acids holding together the NAGs and the NAMs in peptidoglycan, right? The name peptidoglycan, what does it sound like? Which part sounds like glucose? Glycan sounds like glucose. And then which part sounds like, in the beginning, peptido, that sounds like peptide bond, yeah? So these amino acids in the little cross-bridge amino acids and side chain amino acids are used to link the basic structure of the carbohydrates one to another. Is that good? We'll talk more about peptidoglycan structure later on, but this is some more information that should be useful to you. Okay, moving on. All right, proteins are composed of thousands of amino acids, or hundreds. arranged in specific linear sequence. And proteins owe their diversity, complexity, and versatility to the linear sequence arrangement of the 20 different amino acids of which they are composed. You understand what I'm saying there? So, proteins are incredibly complex, as I've said before. They're molecules, but they're about the most complicated molecules around. They're also incredibly diverse, right? Your muscle proteins are way different from your blood proteins, are way different from your hair proteins, right? And that's because they have different arrangements of 20 different amino acids. Instead of being made out of one monomer, like starch, right? Starch is nothing but glucose over and over, right? Cellulose, nothing but glucose over and over, right? Peptidoglycan, nothing but NAG and NAM over and over, yeah? Amino acids, there's 20 different amino acids, and you can throw any particular amino acid into your train that you want. Okay, is that clear? How complex can that be? How many possibilities are there? How many amino acids? 20 different amino acids, yeah? So if I have one amino acid, how many choices is that? One amino acid. 20 choices, yeah? What if I have two amino acids linked together? Like that. One, there's 20 different possibilities for the first one, 20 different possibilities for the second one. How many dipeptides exist in the world? How many possible dipeptides can exist? Heard a couple answers there. What's the right answer? 400 is the correct answer, yeah? Yeah, so 20 times 20. That's for two. So one amino acid, 20 choices. Two amino acids linked together, 400 choices, yeah? Three amino acids linked together. How many choices then? 8,000, right? 400 times 20, yeah? So three amino acids, we're already up to 8,000 choices. What about four amino acids? 160, is it 160,000, yeah? And then 5, 3.2 million, yeah? So 5 amino acids, we're already up to 3.2 million possibilities. Does that make sense? But we don't have 5 amino acids in a protein. You have 5 amino acids, you have what's called an oligopeptide, right? You need 300 amino acids to make a typical protein, yeah? So the numbers are off the hook, yeah? They're virtually limitless. Does that make sense? They're so limitless that a computer is not going to solve them for you, okay? How do you make, so how do you make a protein? Well, the code for a protein is on DNA. If you know that DNA is important, the reason it's important, that's correct. DNA is important, but the reason it's important is because it's the blueprint to make proteins, and you are made out of proteins. Does that make sense? If your DNA gets messed up, what? Your proteins are messed up. Does that make sense? Okay. So, how many people have seen something like this before? If you have, good, this is important. If you haven't, take a good look. Try to file it away in your brain somewhere. You'll see it again. Okay? Is that good? All right. So one thing to note is that, again, amino acids are linked in a chain. And we have little purple balls. Those are our amino acids. And they're in a chain just like rail cars, yeah? Okay. So how do you get all these shapes, right? All those pictures of squiggles, of linear squiggles to make the complex shapes of proteins, where do those pictures come from? Well, they're not so easy to get. Each one of those pictures that I showed of ATP synthase and fibrin and so on was a heck of a lot of work for people who have jobs as what's called an X-ray crystallographer. So basically what you do is you take a bunch of the protein, you purify it, and then you dehydrate it and get it all dried out until it forms a little crystal, yeah? And that crystal, you shoot a laser at it, and when you shoot an X-ray laser, a super powerful X-ray laser at it, then you see a little shadow print here, yeah? So what's this? Ruff, 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 ruff. What is it? It's not really a dog, right? It's my hand, right? You're not fooled, right? It's my hand, right? But if you only saw the shadow, you would still not be fooled. You would still know it was my hand. Does that make sense? By looking at the shadow, you could have figured out what it was, yeah? And I had the opportunity to do this in front of the class, yes? Likewise, by looking at this shadow print, we can infer the shape of this thing that casts the shadow. Does that make sense? All right, and that job is hard. I don't see how, looking at this, that looks like a hard job to me. In any case, once that shape is determined, right, that's what we get. Yeah, so that's how we get those pictures, most of them. However, when proteins cannot be crystallized, you can't get a computer to predict their shape. The numbers of possibilities, right? We talked about 500 amino acids and the number of possibilities there. That number is beyond the scope of calculation for a computer. Furthermore, we have a three-dimensional object, right? It's a ribbon, but that ribbon scrunches up into special protein structure. And that three-dimensional structure is exponentially even more complex. So it's not just that computers now are not up to the task. Computers are never going to be up to the task of figuring it out. The complexity of proteins is not something that computers are going to figure out. So when proteins can't be crystallized, we'll never get a computer to tell you what the shape is. But we might get this, and this you might find interesting, we might get a teenager to do it. Calculating the shape of a protein is an impossibly complex mathematical problem, but at least for now, humans have one thing that we're better at in terms of cognition than computers, and that is something called pattern recognition and intuition. Humans are more intuitive, and they're better at detecting patterns than computers, at least for now. Okay? So what do? You make a video game, and you get kids to play it. Can I briefly digress about this? I think it's kind of interesting. Okay? So no, don't stop recording. Let's let the folks at home watch or read an article about, let's see, news.google.com. We'll go to Google News, and then we'll go to fold.it, HIV. Folded gamers solve riddle of HIV enzyme within 3 weeks. Continue to cite. Okay, close. Folded gamers. So there is the man who cured HIV. Yay! He's a gamer. Okay. What do you think? Hopefully you give a few more props to the guy who designed the game. Shall we read this? Can you guys read it? Here, I'll make it bigger. When video gamers, armed with the world's most powerful supercomputers, take on science and its most vexing riddles, who wins? Sometimes it's the gamers. Humans retain an edge over computers when complex problems require intuition and leaps of insight rather than brute calculations. Savvy programmers and researchers at the University of Washington, go Huskies, have tapped into this human supercomputer with Foldit, an online game that poses complex puzzles about how proteins fold, one of the hardest and most expensive problems in biology today. Foldit attempts to predict the structure of a protein by taking advantage of humans' puzzle-solving intuitions and having people play competitively to fold the best proteins. Since proteins are part of so many diseases, they can also be part of the cure. Players can design brand new proteins that could help prevent or treat important diseases. So this is design brand new proteins is not what the article is about, yeah? This is about figuring out an existing protein, but proteins can be synthesized synthetically by imagining them. So they've been on a tear ever since with 4 publications and a host of puzzles left to solve from deadly diseases to biofuel. Foldit has proven the concept works. Their latest solution has resolved a problem stumping scientists for a decade. Publishing in the journal Nature, Structure, and Molecular Biology on September 18th, researchers show how gamers provided crucial insights to solve the structure of a protein snipping. This shouldn't be sniping. This should be snipping. It's a typo. Enzyme critical for reproduction of the AIDS virus. So this is a enzyme, a protease that the enzyme or the virus uses to make you sick. Yeah. Why would we care about the shape of this enzyme? Well, once we know the shape of the enzyme, what can we do? We can find ways to mess with it. Does that make sense? And if we can mess with it, then what are we doing? Well, we're messing with the mechanisms of HIV, disease, infection, and transmission, and so on. Yeah? Does that make sense? Is this interesting? I think it's pretty huge, yeah? And hopefully it's instructive, right? It tells you the structure of proteins is complicated, but solving the shape of proteins matters for medicine. Is that clear? So lastly, we're not going to play this game, but fold it. So there it is. Behold, the game Foldit. So you get to download in Windows, Mac, or if you're a real cool dude, Linux. I think people describe Linux as rather nerdy, I guess, yeah. Not that I know a lot about such things, but yeah. So you could probably basically you play with the shape of a protein, and if the protein's more thermodynamically efficient, then you get more points. Is that pretty exciting? I actually assigned people to play it once as a homework assignment, and that wasn't super successful, but yeah, people are like, why am I doing this? So I no longer do that. Anyway, you get the idea? Okay, so 4 levels of protein structure. It's now 2 o'clock, and I think I'm going to call it a day. It's about 5 minutes to 2, and so I'll stop talking now.